During cell division, chromosome attach with spindles. A. Kinetochore, B. Centrosome, C. Centriole, D. Secondary constriction. Due to attachment of spindle fibers at the centromere. Centromere is also known as kinetochore of chromosomes. This is a chromosome, this is the centromere. Spindles attach here. The chromosomes are arranged in the center or at the equator or at the equatorial plate due to active movements. These are centrioles. Spindle fibers are attached to centromere or kinetochore. So option A is the correct answer. Next question is, pattern of organization of cilia and flagella is A 9 plus O, B 9 plus 1, C 9 plus 2, D 9 plus 3. Cilia are fine hair-like protoplasmic extensions of which consist of 9 peripheral axoneme and 2 central molecules. The 9 peripheral are microtubules and 2 central microtubules arrangement are seen in cilia and flagella. These are covered by central sheath. And 9 plus 3 arrangement is seen in centrioles. So option C is the correct answer. Flagella with single strand and composed of flagellin is found in A. prokaryotes, B. eukaryotes, C. both A and B, D. none of this. The single strand flagella occurs in prokaryotes, primitive nucleus organisms like bacteria. They contain only single stranded and does not contain 9 plus 2 system. So option A is the correct answer. So our next question is, peroxisomes do not have A. D-amino acid oxidase, B. beta hydroxy acid oxidase, C. urate oxidase, D. amylase oxidase. Peroxisomes are microbodies which contain enzyme for synthesis of peroxides. Enzymes like oxidases and catalases. So it contains D-amino acid oxidase, urate oxidase and catalase like amylase. It does not contain beta hydroxy acid oxidase. Which of the following are included in the category of microbodies? A. peroxisome, B. spherosome, C. glyoxysomes, D. all of the above. Microbodies take direct part in oxidation of substrate. Peroxisomes forms peroxides and oxidize the organisms. Spherosomes helps in storage and synthesis of fat, mostly seen in plants. Glyoxysomes helps in oxidation of fatty acids. So all of these are included under microbodies. Allelone grains are A. enzyme, B. carbohydrate, C. protein, D. fats. Allelone is a structure present in a seed. It helps as a nutrition to the seed. This is allelone. It is proteinaceous structure and consists of proteins. So option C is the correct answer. High content of lysine is present in A. wheat, B. apple, C. maize, D. banana. Lysine is an essential amino acid. It is found in wheat and it is not present in human body. It is not synthesized in human body, so it should be taken as supplement, so it is known as essential amino acid. Option A is the correct answer. Which of the following is the simplest amino acid? A. tyrosine, B. asparagine, C. glycine, D. alanine. Glycine is considered as the simplest amino acid as it possesses only one amino group and one carboxyl group. and neither substituent or functional group is present. So option C, glycine, is the simplest amino acid. Which of the following amino acid is not optically active? A. Glycine, B. Valine, C. Leucine, D. Isoleucine. Glycine is not optically active because it is simplest amino acid. It consists of only one amino group and one carboxyl group. It does not contain any substrate or other functional group. So option A is the correct answer. The telomere of eukaryotic chromosome consists of short sequence of A. Adenine rich repeats, B. Guanine rich repeats, C. Thymine rich repeats, D. Cytosine rich repeats. Eukaryotic telomeric DNA has been shown to consist of simple randomly repeated sequence characterized by cluster of G residues in one strand and C residues in other. Another feature is 3 overhang of the G-rich strand. These overhangs are 12 to 16 nucleotides in length. So option B is the correct answer. DNA is a polymer of A nucleotide, B nucleoside, C amino acid, D all of the above. DNA is double helix structure formed of one nitrogen base, maybe adenine, guanine, cytosine, or thymine. Uracil is found in RNA. So it consists of nitrogen base, one sugar molecule, which is deoxy sugar, and one phosphate. All these attaches to form nucleotide. When this nucleotide repeats itself in DNA, so it is a polymer of nucleotide. Option A. The strongest evidence that DNA is a genetic material comes from that A. that chromosomes are made up of DNA, B. transformation of bacteria cell, C. that DNA is not present in cytoplasm, D. that DNA is present in nucleus. So the evidence that DNA is genetic material came from transformation of bacterial cell. It was experiment seen in by pneumonia bacteria and rat experiment. So it was transformation change. Option B. Strand of DNA are bonded by A. hydrogen, B. carbon, C. oxygen, D. nitrogen. There are double bond hydrogen between adenine and thymine and triple bond between cytosine and guanine. So these are hydrogen bond present between DNA. So option A. RNA contains A. hexose sugar, B. deoxyribose sugar, C. dextrose sugar, D. ribose sugar. RNA consists of sugar, which is ribose, 5 carbon sugar attached to nitrogen base and phosphate. The nitrogen base thymine is replaced by uracil in RNA. Deoxyribose sugar is present in DNA. So option D is the correct answer. The hydrolysis of nucleic acid yields A. only sugar, B. phosphoric acid only, C. nitrogen base only, D. all of the above. A nucleic acid, also known as nucleotide, hydrolysis of this yields a phosphorus, which is attached to a nitrogen base, which is attached to a sugar. This forms a nucleotide. On hydrolysis we get all of this. Recombination of genes occurs at A. prophase in mitosis, B. prophase 1 in meiosis, C. prophase 2 in meiosis, D. metaphase 2 in meiosis. Crossing over of homologous chromosomes occur in prophase 1 of packetine. It is also known as recombination. Crossing over. So option B is the correct answer. The following position is the base to define the formation of place of cell plate at the end of the nuclear division. A. microtubule, B. vesicle, C. endoplasmic reticulum, D. Golgi bodies. Vesicles provided by Golgi apparatus to form phagoplast, phagmoplast, which join to form cell plate. This cell plate is first laid down at the center and then proceeds towards periphery. So option B is the correct answer. Study of nuclear cytology is called A. Neurology, B. Karyology, C. Mycology, D. Rhinology. Karyo means nucleus, and nuclear study or nuclear cytology is known as karyology. Neurology is the study of neurons of central nervous system, basically CNS. Mycology is the study of fungus. Rhinology is the study related to nose. So option B is the correct answer. The term nucleolus was coined by A. R. Brown, C. H. Hooks, C. Bowman, D. Hastings. Nucleolus was discovered by Fontana, and the term nucleolus was given by Bowman. So option C is the correct answer. Robert Brown was the one who discovered and named nucleus. Karyolymph is A. Nuclear sap, B. SPM membrane, C. Nuclear pore, D. None of this. Colloidal sap inside nucleus is known as nucleoplasm or karyolymph. So it is nuclear sap, option A. Which of the following is present in mitochondria? A. Polysomes, B. Monosomes, C. Quantasomes, D. Oxysomes. Oxysomes or elementary particles or F1 particles. These are responsible for respiration or respiratory chain phosphorylation, also known as oxidative phosphorylation. So option D is the correct answer. Right for cellular respiration is A. Nucleus, B. Ribosome, C. Mitochondria, D. Endoplasmic reticulum. Cellular respiration takes place inside the mitochondria. It is a double membrane structure. Inner membrane folds into various cisternae. Mitochondria has F1 particles, also known as elementary particle. It has base, this is base, stalk and headpiece. This headpiece consists of various enzymes for oxidative phosphorylation. So cellular respiration takes place in mitochondria. In prokaryotes, mitochondria are absent. Even then Krebs cycle takes place. What is the site of Krebs cycle in bacteria? A. Ribosome, B. Nucleoid, C. Cytoplasm, D. Mesosomes. Infoldings of plasma membranes in bacteria is known as mesosomes. These are analogous to mitochondria. This is the nuclear membrane, and infoldings of the nuclear membrane is known as mesosomes. Site for Krebs cycle. The following cell organelle is considered to be rich in catabolic enzymes. A. Endoplasmic reticulum, B. Lysosome, C. Golgi bodies, D. Mitochondria. Of all this, mitochondria is involved in cellular respiration, which forms ATP. Golgi bodies helps in secretion and storage and formation of cell wall. Endoplasmic reticulum, there are two types. Smooth helps in synthesis of fat and rough helps in synthesis of proteins, RER. Lysosomes consist of enzymes and destroy the particles present in the cell, or at times destroy the own cell organelles. So it is known as autolysis or phagocytosis. So rich in catabolic enzymes is lysozymes, also known as suicidal bags. DNA is present in A. carboxysome, B. ribosome, C. lysosomes, and D. mitochondria and chloroplast. DNA is present in mitochondria and chloroplast. It may be circular or extracellular like plasmid, but it is present in these organelles. Lysosome, ribosomes, and carboxysomes does not contain DNA, and they are non-membrane organisms, cell organelles. So option D is the correct answer.